Hi everyone, I'm Wilhelmina or Billy Solly, and I'm going to be talking to you today about Anderson et al. 2022 and fMRI networks and creativity in Big C artists and scientists. So the research team in this study wanted to build on prior inconsistent research about both brain functionality and connectivity, and comparing creative individuals to the general population. So they wanted to do this by looking at Big C, or exceptionally creative individuals, but use an IQ-matched comparison group, unlike previous research, to eliminate a lot of possible confounding variables. They wanted to look at two domains within creativity, looking at both visual artists and scientists, and to use an fMRI scanner to look at the functional connectivity within the brain during both divergent and convergent thinking, and, also while participants were at rest. They then wanted to use graph theory to calculate both global and local measures of functional connectivity. The primary hypothesis was that the Big C groups would differ from the comparison group and or each other on sigma or modularity. This sigma refers to the small worldness, or little localized pockets of brain functionality with high clustering and short pathways, whereas the secondary hypothesis wanted to look at if group conditions would react differently to different tasks, or if they would use different brain networks. The first thing to do was to get these participants. So they wanted to look at three groups, and the first is the Big C groups, the exceptionally creative individuals. And within this, they wanted to look at visual artists and scientists. The reason for looking at these two domains is that the Creative Achievement Questionnaire has suggested that visual arts and science have distinct creative domains from each other. So while they are both exceptionally creative individuals, they are also separate domains within themselves. And the way they found that these exceptionally creative individuals was having them nominated from others within the field, and then verifying that by seeing how they performed on the Creative Achievement Questionnaire and requiring that they be in the top 2% compared to the general population. They then found their SMART comparison group by getting IQ-matched-ish individuals, and from the UCLA 300 project, who had said they would like to be contacted about future research possibilities, and from people with graduate degrees. And they made an effort to match the SMART comparison group to the Big C groups in terms of age, sex, race, and ethnicity. And in the end, once the analysis was run, none of the groups differed significantly from each other on any demographic information or IQ. Once they had these participants, they were put into the fMRI, measured for 6 minutes at rest, just looking at a screen, and then they were given the AUT and RAT tests. The AUT, or alternate use for common objects, meant that a participant was given an object, like an umbrella, and asked to think of different uses. And for every use they thought of, they would click a button, allowing the researchers to place the brain image in relation to the thought process of the individual. The remote associations tests, each individual was given 3 words and had to think of a word to connect them. And when they thought of the word to connect the 3 words, they would click the button, again allowing the researchers to place the brain image in terms of the thought process. Once these images were collected, they were analyzed for global connectivity, local connectivity, and global and local clustering. So the results showed that the groups did not perform differently on the AUT and or RAT tests, but that their brain functions did during the tests. So the global connectivity analysis showed that the scientist group specifically had reduced sigma small world connectivity compared to the smart comparison group. This means that their thinking was less in those localized pockets with short stems or short pathways and a little more broad in terms of the brain area covered, but that the visual artists were kind of in the middle of those two groups and not significantly different. The local connectivity analysis showed that the big C groups both had higher local efficiency while they were resting, so their brain was a little bit more localized. But during the AUT test, that went down, so it was a little less local and efficient and more broad and spread out, but that there was not a significant difference during the RAT test. Then looking at the global and local clustering, during REST, global clusters were anti-correlated with local clusters in all of the groups. So these results indicate that reduced sigma may be indicative of higher levels of creativity, and that the high global connectivity could show that big C individuals are able to link separate regions of the brain together compared to non-big C individuals. Both of these show that maybe a more global view of the brain and being able to use it less localized could be indicative of higher levels of creativity in individuals. This also suggests that the AUT test might be less demanding on big C individuals, and that it might just be a little more chill for them to make those connections. And it is also possible that increase in local connectivity and those short pathways and pockets could come at the expense of global connectivity because of that anti-correlation that we saw in the results section. These findings, I think part of the importance comes from the fact that in the discussion they mentioned that some of the brain characteristics of these big C individuals are actually similar to brain characteristics found in people with diagnosed mental illness such as ADHD and schizophrenia, which is really interesting if you think about societally, if we find these differences and we find them indicative of something like a mental illness, and then they are found in children or in people who aren't diagnosed, people could view that very negatively. They could see a lot of problems with it, versus now we're looking at those same characteristics of the less localized thinking and applying it to these creative individuals that people generally have a very high view of and are impressed by. And I think finding these creative, these differences in both of these places, but having the same brain structure, is really interesting, and I think it's important to be able to continue this research on the brain and study with both in a positive and negative light, and not necessarily making value judgments on it and on how brain structures work, and using that to show that, even though brains can be different between individuals, there's not necessarily a good or bad or a right or wrong, and I think that furthering that research into many different groups could be really helpful and important for more and more people to learn about. So I hope you all enjoyed the presentation. I hope you learned a little bit, and I hope you have a great weekend. Thanks for watching.